We're going to take a look at dihybrid crosses. Dihybrid crosses are when we are considering two traits and how they are inherited together at the same time. Monohybrid crosses are only looking at one trait, so the Punnett squares don't get bigger than a 2 by 2. In this case, maximum we can get up to a 4 by 4, but I'll show you later, you don't actually have to do a 4 by 4 depending on the actual genotypes of the parents. So we're going to try to touch upon a few issues, special issues in meiosis, and hopefully you can see how all of this fits together, how a dihybrid cross works, what its relationship with meiosis is, and Mendel's second law of independent assortment. But we'll use an example. So we'll pick up a few things as we're trying to do this. So what we're going to do is we're going to consider two traits, and the two parents will be pure breeding strains. What that means is they're not going to be heterozygous for their traits. They're going to have two copies of each of the alleles for their particular traits. So we're going to use 4 letters here, and when starting off any type of genetics practice question, of course, you should make sure you assign the alleles. So we're going to let big R equal round and little r equal wrinkled. Round is dominant over wrinkled based on the big letter and the small letter. And then the other trait that we're considering at the same time is color. So this is seed shape, and this is going to be seed color. So big Y means yellow and little y means green. If you're writing this by hand, make sure that your big Y and your little y look significantly different. So we're going to have two parents. One parent is going to be pure breeding for the two dominant traits, in other words, round and yellow. So one parent is going to be round and yellow, and the other parent is going to be pure breeding for the recessive traits, which are wrinkled and green. So there it is, and let's add the words there, green and wrinkled. So always start with the phenotype and then the genotypes. Once you've got all of that, that's the hardest part, and you can move on. So the other phenotype for this parent is round and yellow. Is that too small for you to see? I can enlarge that a little bit. So I said they're pure breeding, so that means they're not going to be heterozygous for the trait. So the pure breeding parent that is round and yellow will have two copies that make it round, two copies of the allele that make it round, big R, and then two copies that make it yellow, big Y, big Y. So that makes sense like that. You keep the alleles for the same gene together, so we're not going to have big R, big Y, big R, big Y. It'll just make it a lot easier to keep track of. And the green wrinkled parent, the genotype will be pure breeding, little r, little y, little y. Every parent, every individual, here's an individual, has two copies because this individual got one copy, one big R from one parent, another big R from another parent, and a big Y from one parent, big Y from another parent. Over here, very large size, you can see these are diagrams of the chromosomes. So this actually represents this parent here, and this actually represents this parent here. These are the chromosomes that they inherited, this one from the dad, this one from the mom, this one from dad, this one from mom, just for simplicity's sake. And you can see they have big R, big Y, big Y, little r, little y, little y. So this is what it would look like in meiosis, for example. Now we're going to look at the possible gametes that we can get. For the gametes, based on Mendel's law of segregation, we can only pass one copy into the gamete. So I can only give one big R and one big Y. So you should be able to see, and now I'll illustrate this in meiosis, when we're actually in gamete formation, during anaphase, how this lines up here can create different variations. But you can see here, no matter how this lines up, even if this lines up over here, or if there's any kind of crossing over happening, because they're the same allele, you're not going to get any new variations. The only combination you can get is actually a big R and a big Y. That's the only possible gamete. Pretty easy to solve. And the same situation for this parent here, no matter how crossing over happens, or this would be crossing over here, or random assortment would be like this. No matter how they line up, you're only going to end up with one possible gamete configuration over here, and that is little r, little y. So it's very easy for me to figure out what the kids are going to be. Now if I combine these two, I'm going to get big R, big Y, little y. All the kids will look exactly the same. They're going to be big R, they're going to have this genotype, and what phenotype does this spell out? Well, it actually still looks like this parent because of the dominant R and the dominant Y, which are round and yellow. So the kids will look like this. Now it gets more interesting when we try to make the kids have babies with each other. Now this would be wrong for humans, right? But for pea plants, that's totally fine. So we're gonna do that. So here we go. We're gonna do the same thing. Let's start with having two of these babies. Okay, here's one of them, and it's gonna mate with another one, and their genotypes are heterozygous for both traits. Now this is going to be a little more interesting. So the genotypes are heterozygous for both traits. I just grabbed that from up here. Now when we look at the possible gametes that can form, if I scroll down a little bit, can you take a look? Here is the situation. Let's ignore all this over here. Just look over here on the left. So this is what these babies inherit. They inherited a big R from mom or dad, and then a little r from the other parent. Same thing here. Now look at what can happen if this lines up in a different way. So this is the equator. So if anaphase were to happen, this big R could go with this big Y. That's one possible gamete. This little r could go with this little y. That's another possible gamete. But if they happen to line up like this, and we can expect that to happen 50% of the time, now I can have another possible gamete, big R and little y, and then I can have a little r and big Y. So did you count up? That's 4 possibilities. 4 possibilities. So if I wanted to show it here, you don't have to draw out chromosomes every single time. A big R and a big Y can come down together. A big R and a little y. That's one other possible gamete. And I like to put my gametes in circles. A little r and a big Y. And a little r and a little y. There's only 4 possibilities. Remember, you cannot inherit both copies of the same allele. So you can't form one, for example, that's called big Y, little y. You have to have one of these two combined with one of these two. And these are the 4 possibilities. So if there are your possible gametes there, and this is the most famous example, by the way, so you're going to really recognize this. Same thing over here. These are the 4 possible gametes. Now look at this. Let's add the phenotype here, round yellow. I think we know that, round yellow. If we do a Punnett square out of this is going to be a 4x4 Punnett square, which I've kind of prepared down here. You should actually try to do this by hand and write it all out, at least once, just to make sure you're doing everything correctly and see if everything adds up correctly. So it turns out if I do all of this and I'm going to write, here's the Punnett square. These are the 4 gametes from one parent. 4 gametes from the other parent. Remember, this is the second generation. The first generation was all the way up here. This is the second generation, so we're breeding the second generation with each other. And just to save some time, I've already done the entire thing. Now, take note of a few things here. You keep the R's together and you keep the Y's together. You put the capital letters first and then the little letters afterwards, just for consistency's sake. And it'll help you when you're adding all of these up. So it turns out, this is why you should try this by hand once, just to make sure you're doing it all correctly. Once I do it all, I've actually used some color to categorize these. It turns out all of these with this rounded yellow highlighter type thing have the characteristics of round and yellow, right? Big R, big Y is present in all of them, round and yellow. These ones that are green are round and green, round and green. These ones that are in a cross out right here are wrinkled and yellow. Little r, they're all homozygous recessive for little r, wrinkled and yellow. And there's only one that has both of the recessive characteristics. So out of this Punnett square, if I were to do a probability graph, this is what I would expect. I would expect that out of 16 kids, of course you wouldn't have exactly 16 kids, so you have to do a ratio. Out of the 16 kids, the ratio turns out to be you get 9 of these round yellow, so both of them show both of the dominant characteristics. You get 3 showing round and green, which is a dominant characteristic with a recessive characteristic. 3 with the opposite configuration, a recessive characteristic and a dominant for the color, and then 1 showing both of the recessive characteristics. If you do all of that, look at that. 9 to 3 to 3 to 1. That is a famous ratio that you will get. It turns out that this is the ratio that you always get for this exact situation where you are mating 2 individuals that are both heterozygous for 2 traits. Doesn't matter what letters you use, big A, little a, big B, little b, big A, little a, big B, little b, you will end up with this situation 9 to 3 to 3 to 1. So you should definitely take note of that and always recognize 9 to 3 to 3 to 1. Now, a few other things that I wanted to point out. We're going to learn about gene linkage in a later video. For now, you should take note that this situation right now, this box is quite important. What we have just done, we've done a sample genetic cross, a dihybrid cross. We are assuming that the genes are unlinked. That means R, the gene R controlling seed shape, and the gene Y controlling seed color are on different chromosomes. And because they're on different chromosomes, I can actually have an equal probability of these things lining up. Remember, in this parent, it doesn't make a difference. But if I scroll down, in this parent situation, it does make a difference. So one more time, back up to the box. Please write this down. We have just done an example of two unlinked genes. Unlinked means they are on different chromosomes, and just for note-taking purposes, these are autosomal genes as well too. They are not the sex chromosomes. And we don't have to do any sex chromosome dihybrid crosses. I thought it would get very, very complicated and probably would only exist for linked genes. So what we've just done, unlinked autosomal genes showing pure breeding parents creating heterozygous for two traits child. We link both of these together. We have two of those children make babies with each other. They are both heterozygous for two traits, and we get this predicted 9:3 to 3:1 ratio. And I've shown you how by rearranging how they line up here during metaphase, you can actually create 4 possible gametes. That's why you have to do a 4x4 Punnett square. It's called random orientation when they line up. If it lines up like this or lines up like this, that leads to lots more variety. If you consider this is actually only 2 of the pairs, there are actually 23 pairs of chromosomes. And just a reminder, Mendel's law of independent assortment states that the allele pairs separate independently during the formation of gametes. So what Mendel is basically saying is that R is not affecting Y. This is just as likely to happen as this is just likely to happen. So we would expect equal amounts of these possible gametes, which makes this a predicted ratio, perfect for things like the chi-squared test. I want to show, this is just one really quick example. If you're given something like this, where one parent is heterozygous for both traits and the other is wrinkled and green, which would make, they'd have to be homozygous recessive for too. So I'm going to do this really, really fast. The possible gametes here, as soon as I see an individual that is heterozygous for both traits, then I know that there are 4 possible gametes. This only 1 possible gamete. So check it out. If I were to do a Punnett square for this, it's only a 1x4 Punnett square. Oh, I brought everything too low. So I'm just gonna move these guys up here. Here are our 4. Oops. Here's our Punnett square that we're making. And if we actually combine all of this together, put in the actual genotypes, let me check that it's right. Big R, little r, big Y, little y, big R, little y, little y, little r, big Y, little y, little r, little y, little y. And take a look at that. Every single one of these kids is going to be different. So in this case, it's a 1:1:1:1 ratio. Why is it not 9:3 to 3:1? Because both parents are not heterozygous for both traits. So if you don't have a situation where both parents are heterozygous for both traits, then you can actually do a very quick Punnett square. It's more likely on an exam that you'll be given a situation like this, because writing all of this out in an exam would take a lot of time. You'll be lucky if you get it because you know how to do it now, but more importantly, be able to recognize as soon as you see 2 parents heterozygous for 2 traits on unlinked autosomal genes, you're always going to get a 9:3 to 3:1 ratio. Make sure you understand this, because when we discuss gene linkage in a future video, you really need to understand how this is the main rule, but it can be broken when the genes are actually linked together. Okay, sorry that went on a little bit too long. Hopefully you understand that a little bit. I've got another part which I'll set together as a part 2 for this.